We have no explanation. Things like this just happen. He has autism. He has very severe autism. At this point, Andrew's 4 years and 9 months old. He didn't like to really play with toys. He would pull the books off the bookshelves or VCR tapes off and just play with them on the floor, but not really in a meaningful way. He was very stiff and postured. A lot of toe walking, which is seen in children with autism. Of course, he's not really making eye contact here at all. He had no language at this point, just he uttered noises. Mostly he uttered E, E, over and over again. He seemed healthy, except for, and this is very important, he had a lot of GI symptoms. His stool was foamy, mucousy, bulky. It wasn't like anything I ever seen come out of a kid before, and with 4 kids. You know. He started to refuse to eat solid food, but he would eat non-edibles, tissue, paper, ashes from the fireplace, the drywall off the walls.
Ellen's biggest fear was that the developmental window for Andy to acquire language was quickly passing. She had to do something.
I felt called to start researching in spite of having no medical background at all.
Ellen's starting point was Andy's history, compared with her first 3 children's development. The only difference she found with Andy was his experience with antibiotics. It didn't take long for her to stumble upon the name Feingold.
One of the lesser-known problems with antibiotic use is that they do impact the bowel flora.
At 90 years old, Dr. Sidney Feingold is still considered one of North America's best microbiologists. He's been researching bowel flora since the 1950s.
The human bowel is a remarkable organ. If you were to stretch it all out, it would be the dimensions of a tennis court. It has many different bacteria in it normally, a great variety, probably between 1,000 and 10,000 different types of bacteria. These children are susceptible to ear infections, and so they receive antibiotics, and the antibiotics intend to select out certain organisms while they suppress much of the rest of the flora. Now, the organisms that tend to persist are Clostridia.
Clostridia are a family of around 100 species of bacteria. A few of them are pathogenic, extremely hardy, and resistant to most antibiotics. They also produce neurotoxins that affect the brain. But Ellen found that it might be possible to attack the Clostridia specifically with a particular antibiotic.
I really felt, if we go on this premise, if we act on this premise, he can be helped. An antibiotic that specifically targets Clostridium in the intestinal tract could potentially be a huge benefit to him. So I started writing doctors. Many of them simply referred me to somebody else because they felt it was, outside of their expertise. It didn't clearly fit with infectious disease. It didn't clearly fit with gastroenterology. It didn't clearly fit with neurology, immunology. So I was hitting on all of these different specialists.
After 15 months and 37 doctors, Ellen found a gastroenterologist who agreed her theory should be tested. They began Andrew on an 8-week trial of one of the only antibiotics known to kill Clostridia bacteria.
The results were astounding.
Oh, wait.
Within a matter of just a few weeks, he became calm. He was aware of his environment and responsive enough to begin potty training, which was instantly successful.
Good boy!
He's now beginning to play with toys. He's leaving clothing on.
Yes. Yes. That's your new word, huh? Yes. And no. No. Good boy. Good thing. No. Oh, good boy.
He's putting puzzles together.
Does them all backwards. He did his name backwards. He's doing these all backwards.
Oh, nice kisses. More kisses?
That was the end of the 8-week trial. Dr. Sandler on the spot said, well, we need to test this with more children.
At this point, Ellen and her doctor took their story to Sidney Feingold.
So, Julie, you got some black colonies there?
Yes, these are growing nicely actually on this plate here.
And he agreed to help with a pilot study of 11 more children.
How long did they take to show up?
That took 2 weeks to come up.
2 weeks.
Pigmentation, yeah.
I've always felt that bacteria in the gut do things other than cause infections of the classic type. There was never evidence for it, and here was an opportunity where, even based on that one initial patient, Ellen Boldy's son, it seemed very logical that bacteria in the gut played an important role.
Sure enough, Dr. Feingold found Clostridia in higher amounts in the stools of the test children, and that 80% of them improved on the antibiotic vancomycin. But there was a catch. In a hostile environment, Clostridia produced spores, essentially going into hibernation.
Actually, spores are resistant to all known antibiotics. There isn't an antibiotic that I'm aware of that can kill a spore. Now, I was very concerned that even though we had treated the bacterial cells, I knew that spores remained. And I was quite concerned that when we removed the vancomycin, that any spores remaining in the intestinal tract would germinate and would be right back in business, shall we say. And that, sadly, is exactly what happened. So, you know, there was a lot of very important skills that Andrew was able to maintain, but the behavior, the screaming, the irritability, the don't touch me, I don't want to look at you, the walking on his toes again, repetitive behaviors, those pretty much all came back.
Ellen co-authored a paper with several doctors on the results of the study, and in 2000 it was published in the Journal of Child Neurology. The paper didn't cause the stir in the medical community that was expected by its authors. Though several researchers have since replicated the finding that autistic children have higher levels of Clostridia in their gut, doctors have been slow to pick up the gut bacterial link.
I think we're going to have to do quite a bit more to convince people that are not infectious disease trained or trained in microbiology that bacteria can really do these kinds of things and that they can be controlled.
The medical community may also have ignored Ellen and Dr. Feingold's paper because it was thought to be promoting wider use of vancomycin, which is a drug of last resort in the fight against antibiotic-resistant bacteria. But there were other issues at play as well.
When the results of that study were released, it seems like what everybody focused on was the fact that the children regressed. And I acknowledge the children regressed, but that is not the point. Why did that happen? Why did they get better? Could it be a neurotoxin-producing bacteria in the gut that was suppressed by antibiotic treatment? I think that that's a valid question, and I think it's a question that has not been answered in 11 years since we published that paper.
At his lab at the University of Western Ontario, Dr. McFabe is picking up on Ellen and Dr. Feingold's work with his own gut bug theory.
I was aware of the paper by Dr. Sidney Feingold in UCLA showing unique populations of Clostridial bacteria in the digestive tracts of these children with regressive autism.
Dr. McFabe realized that one of the byproducts of Clostridia is a short-chain fatty acid called propionic acid. It's not a neurotoxin, but it does have dramatic effects.
And then in my previous work, we were working with this compound, propionic acid, and we found that for such a simple molecule, it had very widespread effects in the central nervous system, affecting brain development, metabolism, and immune function. And then, you know, we sort of put 2 and 2 together, and we said, hey, these bacteria in the gut, when children eat carbs, ferment the carbs and make propionic acid. And in addition, some of the foods that these children craved, in addition to being high carb, did in fact contain propionic acid as a food preservative.
Propionic acid is labeled on food packaging as E280. It's a commonly used preservative in many bread, cake, and cheese products made and sold throughout Europe, Australia, South Korea, and North America.
So we thought to take propionic acid and give it in small amounts to rodents and see if the behaviors or brain changes looked anything like autism and whether or not these behavioral and brain changes were transient.
Dr. McFabe's control subjects exhibit typical behavior. Rats are highly social animals. They greet by sniffing nose to tail and interact as they explore their surroundings. But when a propionic acid solution is introduced, this all changes.
What we noticed remarkably within 2 minutes of putting this propionic acid in, this animal immediately starts exhibiting some very peculiar behaviors. Obviously, he's hyperactive and moving very quickly, and then repetitive behaviors. Here you see an abnormal posture of walking with a hunchback. And it's an unappreciated part of autism in that patients often have toe walking, abnormal posturing, arched back. So now we'll place another animal that's been given propionic acid. And a peculiar thing happens. They show none of the normal social greetings of two rodents seeing each other.
A video tracking system records and compares the obsessive and antisocial PPA-treated rats with the normal rats. Other experiments showed responses such as rigidity and object fixation, and the intriguing fact that the effects of PPA wear off.
So now we're about a half an hour after we've exposed these two animals to propionic acids, and their behavior afterwards reverts back to normal, and it's a fat, so it's burned through metabolism. The animals become more social. Usually after their escapades, they tend to just go back to sleep. So again, we have a reversibility of hyperactivity and a reversibility of social behavior.
The autistic-like behavior of PPA-treated rats was compelling. But Dr. McFabe and his team wanted to know exactly what PPA was doing to the brain.
So do you have the lipids on the PPA rats?
Yes, here it is. This is what the final product looked like. When I finish this analysis, we'll be able to quantify whether or not there's any alteration in any of these components.
Brain fat composition is intertwined with behavior. So Dr. McFabe is working with lipids expert Dr. Ray Thomas, who's been examining PPA's effect on lipids, the fats that fuel brain cells.
What we've found is that when we give propionic acid, which is a metabolite from enteric bacteria, it has the propensity or the potential to alter brain fats. They alter the omega-3s, the omega-6, the saturate level. And these alterations that we've found in the brain is closely related or similar to what has been reported in the blood of autistic patients. What we've shown is that with propionic acid, you get a fast change.
Remember, families are saying, my child gets worse with something associated with gut or diet. And sometimes when I make manipulations with diet or gut, my child gets better. We wanted a model that gets worse and better. And what Ray found with these amazingly specific changes in very key lipids that have to do with brain development, brain functioning, and brain energy metabolism. Imagine these gut bacteria produce the propionic acid as a metabolite. It'll fly through all bloodstreams to multiple cells, like all the other fats. But then when it comes to the nervous system, it can get in because it's a small molecule. But there are actually very specific compounds called transporters that actually grab the propionic acid and kind of suck it into the nervous system.
Once in the brain, PPA changes brain fat composition to be similar to that of autistic patients. The brain is 60% fat, and cell function and our behavior are directly related to how those fats are composed. And there's more. PPA could even be switching the genes associated with autism on and off.
Imagine this little molecule from your gut going directly into the nucleus where all your DNA is and acting as a switch to turn on and off important genes having to do with brain development, learning, and memory and metabolism. It's very intriguing that such a simple molecule can do such specific things in a brain metabolism and function that are very consistent with found in autism.
So far, there are no studies showing that PPA or E280 isn't safe for adults when used as a preservative. But Dr. McFabe's work with animals shows that gut bug metabolites like PPA could be triggering autism in susceptible children.
Normal or near normal people exposed to high levels of alcohol are not behaving as their normal selves. And by analogy, I think normal or near normal children exposed to high levels of propionic acid, gut bug metabolites, are not themselves. By removing the propionic acid, by either getting rid of the bug that makes it, not feeding the bugs that are there, healing the gut, or using compounds that help break it down, that will be the approach of treating this disease.
Yes?
Oh, pop, pop.
Oh no.
It's painful, to be honest.
Get them bubbles.
It's painful to watch it because we were so close. I really think that we were so close a decade ago to fixing this, but no one would listen.
Many parents have taken up the gut bacteria theory and are using it to devise treatments.
Andrew is now 19 years old. It's hard to believe that it's been that many years. I do think that it's a war in Andrew's intestinal tract. There is a colony of, I believe, neurotoxin-producing bacteria that established itself and set up camp and set it up good and strong and at an early age. And basically what you become colonized with as a child, those are the bacteria you die with.
Keeping Andy on powerful antibiotics indefinitely wasn't an option. Long-term use would have dangerous side effects, so Ellen turned to other therapies. She has found that by giving Andy massive amounts of probiotics, supplements containing benign bacteria, his symptoms are controlled. So even if Andy's Clostridia can't be eradicated, they can be beaten back with waves of competing bacteria. Studies have confirmed that probiotics have helped other children as well.
He functions very well, and that's a relative term. When I compare